Good afternoon and welcome to the fourth session of our interesting conference. We are now going to talk about evolutionary management of forest. My name is Markus Linder. I'm with the European Forest Institute in Bonn. And I'm chairing the session together with Barbara Vincenti from Bioversity in Rome. And we are going to talk to how the consideration of evolutionary processes can or should be integrated into management and conservation practices. We will have again 7 oral presentations, but before we will start with the keynote presentation, and Barbara is going to announce the presenter, who is not going to be with us, but talk to us through the video.
So good afternoon. I'm going to introduce Sally Aitken, who is giving the next keynote speech. I think I don't need to say much, but just to give a proper introduction. For those who don't know her, Sally is a professor and associate dean in the Faculty of Forestry at the University of British Columbia and the director of the Center for Forest Conservation Genetics. Her research integrates phenotypic, genomic, and climate data to understand the processes driving local adaptation to climate in trees. And she has led recently 2 large multi-institutional applied research projects funded by Genome Canada, AdapTree and co-AdapTree, focused on developing genomic approaches and tools to guide reforestation and breeding for new climates. So I guess the results she is going to present to us will come mainly from these initiatives. So without further waiting, I will give the voice to the video. And Sally, despite the very early hour, she's available to respond to questions, if there are any, after the presentation that will take about half an hour.
Hello, and thank you very much for the opportunity to give you this presentation long distance. I really appreciate Bruno Fatti and the other organizers recognizing that the carbon costs of me coming all the way from Vancouver to Avignon were too high to justify. However, I am really sorry that I'm not there in person to see all of you and hear more about the great progress that the GenTree project has made. So today I'm going to talk about our data that we've collected over the past decade or so on phenotypic and genomic patterns of variation that relate to climate adaptation. And I'll be comparing phenotypic and genomic data sources. So all of you know that we see widespread local adaptation to conifers in boreal and temperate species. And this photograph just shows the marked differences that we see in Sitka spruce provenances, depending on the provenance, ranging from California to Alaska. And of course, climate is changing. We expect that species ranges will shift, and we expect that adaptation will occur as a result of changing selection pressures. So I'm not going to address migration much in this talk, but I just wanted to share with you a recent study that we did that shows just how slow migration can be. So Sitka spruce is a species that has recolonized the Pacific coast of Western North America from California right up to Alaska since the last glacial maximum. And it reached Kodiak Island, Alaska, about 500 years ago. So it has come around the Gulf of Alaska and arrived at Kodiak Island. And we used this colonization on Kodiak, the Kodiak archipelago, to look at some of the factors affecting the ability of populations to expand and colonize after those first migrants reach. So this was the PhD work of Joanne Elouette. Here we have trees that have spread across Kodiak, showing what the leading edge of that range expansion looks like. We've got large trees that are early colonizers still present. There's been little mortality, or there's little evidence of mortality on this archipelago, which make it a great place to go and study both the genetics and the demographics of this species. So there's Joanne taking tree cores. So what Joanne found by genotyping trees and aging them using the tree cores was that there was a pronounced lag in the expansion of populations on the Kodiak archipelago after the initial colonization event. The white bars in the top graph indicate the distribution of trees by age class in Seward, Alaska, where populations have been for a lot longer. And the gray bars indicate the distribution of age classes on the Kodiak archipelago. And corresponding with this lag in population expansion is a lag in the development of genetic diversity, or the buildup of genetic diversity. So the lower graph shows the number of alleles. This is based on RADseq, and shows this delay in the buildup of genetic diversity until about 1700, when both genetic diversity increased and the population really started expanding. I think this says something about how slow the process, the natural process of migration, will be. But what I'd like to talk about mostly today is adaptation in areas where populations already exist. And here we're mostly looking at adaptation to climate. So I'll talk first of all about phenotypic data we have on adaptation to climate. I'll talk about genomic approaches, and particularly how we've used comparative genomics to understand local adaptation to climate. And I'll talk about some recent results we have that are really comparing the information that we get from genomic data versus phenotypic data for understanding climate adaptation. And at the end, I'll talk about some other work associated with our larger co-Adaptree project around both the population genomics of fungal pathogens and also the acceptance of the public and of experts around climate-informed reforestation policies. So most of the results I'll be talking about today come from two large-scale applied research projects, the first being Adaptree, the second being co-Adaptree. We are nearing the end of co-Adaptree now, but we're still mining data from Adaptree, and you can see the project teams here are quite large. This would appear that we were a happier group in Adaptree than in co-Adaptree, but I'm not sure that's the case. But I want to recognize my co-project leaders on these two projects, Andreas Hammond, Sam Yeaman, and Rashar Hamlin, and all of the people who have contributed. So Adaptree focused on lodgepole pine and on interior spruce. Lodgepole pine is a very widespread conifer in Western North America. The interior spruce complex is a hybrid complex consisting of mainly of white spruce, Picea glauca, and Engelmann spruce, Picea engelmannii. And over much of British Columbia, the populations are heavily introgressed. So what does the phenotypic data we've collected on lodgepole pine and interior spruce tell us? Well, the first message is that adaptation to low temperatures really dominates the signals. The map on the left here shows the distribution of cold hardiness phenotypes from a common garden experiment and artificial freeze tests, with dark blue being cold hardy and red being not cold hardy. And next to that, you can see the pattern of 30-year extreme minimum temperatures across the same area. This is across Western Canada, the provinces of British Columbia and Alberta. And when we look at interior spruce, we see the same pattern. Here we've got about 280 populations of lodgepole pine, about 250 of interior spruce. We see very similar geographic patterns reflecting that adaptation to very low temperatures. However, with both lodgepole pine and with interior spruce, we were unable to detect population differences, significant population differences in DEL C13, the stable carbon isotope ratios, or in visible injury from drought, which was a curious thing. So Douglas fir, we've got ongoing work, PhD research of Rafael Ribeiro here, and he's been studying 87 populations of Douglas fir from both the coastal variety Menziesii and the interior variety Glauca, and he also sees, no surprise, strong local adaptation to cold. So we see strong differentiation between the interior variety Glauca and the coastal variety Menziesii here, but we also see a clear latitudinal decline in the coastal variety. And this does not tell us too much about the elevational patterns. So these maps that just show latitude and longitude are not picking up all of the adaptation. So what about drought tolerance in Douglas fir? Well, Rafael has completed two quite impressive experiments looking at drought hardiness, and here you see the progression of one of those experiments. This one contained all 87 populations that he was testing, and you can see the progression of visible injury as well as the control boxes that are remaining green throughout this severe drought experiment. And Rafael and his assistants measured chlorophyll fluorescence all night for many nights to pick up the patterns of how injured these trees were. And on the top, you can see that the wet treatment had consistently high levels of chlorophyll fluorescence, but the drought treatment dropped off over time, and you see that decline in chlorophyll fluorescence. You can also see a clear difference here between the purple lines, which are the interior variety of Douglas fir, and the green lines, which are the coastal variety. So what does that look like geographically? Well, interestingly, we do see, and this isn't a surprise either, we do see quite a strong difference in drought hardiness between interior Douglas fir and coastal Douglas fir varieties. However, within varieties, Rafael did not find significant differences for drought hardiness. And why might that be the case? Well, we might not see population differentiation because of the spatial scale of variability. Water availability varies on a very localized scale, and we were sampling over very broad precipitation gradients. Or it could be that we're not phenotyping the right traits. Drought is extremely complex, much more difficult to phenotype than for cold hardiness, so that may be an issue. But it may also be that these species are globally fairly drought tolerant, but don't vary locally among populations. Now, these 87 populations shown here have all been, we've collected DNA from all the seedlings across our experiments, and we've pooled those samples, normalized libraries, and done exome capture of about 50 megabases. In those, we have the data, and we know we have about 10 million SNPs, but we haven't yet analyzed those data. Another thing we're doing on the phenotypic side is using a case control approach common to medicine. So in this case, with the drought experiments, and we're also doing this, we have done this already for the cold hardiness experiments, what we did is for each of 20 populations, we ran these experiments. We picked the 10 worst trees out of about 80 in each population, and we picked the 10 trees that fared the best under the treatment, and we have pooled each of those phenotypic groups. Those have also been exome captured on pooled samples of those phenotypic groups. And I should just say that the pool-seq approach has given us quite good estimates of allele frequencies in those populations. So we ran tests in Douglas fir, but also in 3 other species, and got quite good results with the pool-seq approach. So let's move into some data that's already been analyzed on the genomic side. And here, the first thing I'd like to talk about, and you may already be familiar with this, but is the comparative approach that was developed by Sam Yeaman and others on our Adaptation project, where we used we used an approach that sidestepped our issue of controlling for population structure to detect local adaptation. So in this case, we have about 23,000 genes for both pine and spruce, and we've got over 1 million SNPs in each species, and we've got over 250 populations. So the same materials I was talking about previously. And again, this work, big credit goes to Sam Yeaman and Kay Hodgins for this. And so what we did in this case was we used both genotype environment associations and genotype phenotype associations to identify, first of all, candidate SNPs that are associated either with phenotypes or with environmental factors. And again, the overwhelming message from these was that cold adaptation and SNPs associated with cold hardiness provided us with the strongest signal. So for this work, we developed a comparative approach where we identified top candidate genes based on the proportion of SNPs across a given locus that were outliers either for phenotype associations or for associations with environment. And these became our top candidate genes. And so here we're looking at correlations without any adjustment for population structure and focused again just on adaptation to low temperatures, and we found about 260 candidate genes in pine and 450 top candidates in spruce. Now these will include false positives, so these are certainly overestimates because they have not been controlled for population structure. But by comparing the two lists, we were able to identify 47 top candidate genes in common between these. And so we are now expanding this approach to additional pairs of species. So we are including in co-Adaptree Douglas fir and western larch. This is a very interesting comparison. These are sister genera separated by about 80 million years of evolution. But of course, Larix is deciduous and Pseudotsuga is evergreen. And then the other pair is a very close sister species pair, which is lodgepole pine and jack pine. And all of these will be compared with our previous results with spruce as well. So, genomic data can be generated fairly quickly, and we can use these approaches of genotype environment associations to look for climate adaptation, or we can look again for associations with key climatic phenotypic traits. So how do the two results compare? If we're trying to make reforestation decisions, for example, assisted gene flow decisions based on data, will we make the same sort of decisions if we're using phenotypic data or if we're using genomic data? So we decided to take a look at this with our lodgepole pine data from Adaptree, and this is work led by Colin Mauney, using a lot of data that was collected by Ian McLaughlin. And you've already heard, I believe, a talk from Ian about his work using some of the same phenotypic and genomic data to compare selectively bred with natural populations. And so here we had, we genotyped these trees with a SNP array. About 32,000 SNPs produced high quality genotypes. And this included about 18,600 contigs and 10 to 30 trees per population. And this work was just published this month. So, first of all, on the left here you see a graph of our raised nursery bed with Ian's Common Garden experiment in it. On the right, what you see is a plot where the x-axis shows the climatic variation that was explained by common garden phenotypes, by 4 common garden phenotypes. And the y-axis is the proportion of climatic variation explained by genomic data. And so each data point on this graph is one climate variable. Each point is one variable. And so what we're doing here is asking, do we identify the same climatic drivers of local adaptation when we use genomic data as when we use phenotypic data? And not surprisingly, again, we see that low temperature adaptation shows up very strongly in both of these data sets. Here we have mean coldest month temperature, temperature difference between summer and winter, degree days below zero, mean annual temperature. Interestingly, mean annual precipitation does not show up in the phenotypic data, but it shows up in the genomic data. And this might be an important indicator of some longer-term dynamics or selection events that we don't pick up in the short term. But this correlation, here we've got an R of 0.56, not bad. But when we look at a 20-year provenance trial, we see a much stronger relationship here, a correlation of 0.9. So this is the Illingworth Lodgepole Pine trial. And so here on the x-axis, we have the climatic variance explained by variation in height in that trial, which is the data that has been collected over time. And on the y-axis, again, we've got climatic variance explained by genomic data, and we see a very strong relationship. Again, the low temperature variable showing up very strongly. Summer moisture variables, cumulative moisture deficit, summer heat moisture index, are showing up in the genomic data, but not in the phenotypic data. And they're definitely weaker signal than the low temperatures, but there's still a signal there that we should pay attention to. But in general, we're getting good information from these genomic approaches, from the genotype environment association approach in this case. So that's good news. So another question that we had was, how is variation distributed across the landscape? So for example, when we have a phenotypic trait and we've got some genetic decline across some environmental gradient, we have one trait and we see some pattern. It might be linear, it might be nonlinear, but we see some pattern across that climatic gradient. Now, when we have genomic data, we have many markers, many genes for most of these traits, and what is the underlying pattern along those environmental gradients? So, for example, do we have largely concordant clines for these different loci or different groups of loci, or do we have more sequential, localized clines? So how is this variation playing out across the landscape? So Colin and others took a look at this by taking cold injury-associated SNPs and running a k-means clustering of those SNPs. And this produced 6 different clusters of SNPs that show similar patterns in response to environmental variables across British Columbia and Alberta. And so here on the right-hand side, we have those 6 clusters plotted, and on the left, they're all plotted together. And you can see that some of those clines are running fairly parallel, but we've got this dark, I want to contrast this dark gray cluster 4 with this light purple cluster 6. Now that top cluster is the only one that's showing much variation at very low temperatures. They're plotted all against mean annual temperature here. So most of these clusters don't vary in the coldest areas. That would be on the far side, the east side of the Rocky Mountains, where temperatures get very low in the boreal. But we've got that cluster 6 that shows a cline in those colder regions. And when we map that, we see, here's maps of the mean frequency of warm associated alleles for cluster 4 and cluster 6 across British Columbia and Alberta. And you can see these distinct patterns of variation that differ on either side of the Rocky Mountains. So the Rocky Mountains run about up the BC-Alberta border for the southern part of this and then head straight on up from there. So we see these quite different patterns. So we have a real mix of patterns spatially. But one good news finding out of all this is that almost all populations were polymorphic for almost all associated SNPs. So even the cold populations had a very low frequency of warm alleles, and the warm populations had a very low frequency of cold alleles, but they were still polymorphic. Okay, so in the last 10 minutes here, I'd like to talk about a couple of other aspects of the Co-Adaptree project. I can't take any credit for these. These are being run by other PIs in our team, but I think they're very interesting from the standpoint of thinking about how we manage forests and how we manage forest management and reforestation decisions in a rapidly changing climate. The first one of these I'd like to talk about is looking at the genomics of the population genomics of pathogenic fungi and also the climate adaptation of those fungi. And this is work led by Richard Hamlin. And then the second thing I'd like to talk about is some work that our social scientists on our Co-Adaptree team have done around the social acceptance and also acceptance by experts of assisted gene flow as a reforestation practice. So first of all, Dothostroma septosporum is a fungal pathogen that causes a disease on pines called Dothostroma needle blight. And this is common around the world on pines, and it can be a big problem under some circumstances. We saw a big outbreak in British Columbia about where these blue dots are here that actually killed quite a few trees about a decade ago. And so one of the things that we built into Co-Adaptree was looking at the genomics of these pathogens themselves. So what you see here is a map of Dothostroma collections in Western North America. This group, our pathology group, has collections actually from around the world that they're also analyzing. But you can see that these fungal accessions fall into different genetic clusters, as are shown in these diagrams on the left, these plots of differentiation. So what this group is doing is whole genome sequencing of these fungi and then analyzing variation within them. But they've also done some work to look at growth rates of these accessions, of a subset of these accessions under different temperatures. So on the left here on this graph, there are 3 temperatures being tested here. 15, 20, 25 degrees Celsius, so the main group's running down, and then each bar there is for one provenance or accession of the fungi. And what you can see is these populations vary quite a bit in terms of their growth rates, but there is also some genotype-environment interaction there with temperature. And these are preliminary data from Richard, Nicolas Faux, and Renata Heinzelmann. So our social science group, consisting of Guillaume Peterson-Saint-Laurent, Shannon Hagerman, and Rob Kozak, conducted surveys to assess public opinion on different reforestation options in a changing climate. And they have done a parallel survey of forestry experts across Canada. The survey of the public was done just in British Columbia. And so the options here were natural regeneration, local tree breeding, assisted gene flow, assisted species migration, non-native species, and genetically modified trees. Now I should say that we do not use any non-native species for forestry in British Columbia, and we do not use any genetically modified trees. So these were put in there as more invasive options, if you will. And the results are quite interesting. Natural regeneration, local tree breeding, and assisted gene flow all had high levels of support, as shown by the green bars. So dark green indicates that people strongly supported a given management practice. A pale green bar indicates that they supported it. And the sum of those is reflected in the percentage on the right-hand side. So 83% of people supported natural regeneration, 90% supported local tree breeding, which was the highest level of support. Assisted gene flow received support of 83%. So there is fair support for assisted gene flow as well as for tree breeding in dealing with climate change. As soon as you talk about moving species outside of their range, support drops. For assisted species migration, that dropped to 53%. Non-native species support was 26%, and genetically modified trees, 25%. Now, it's interesting that the survey across Canada, which just has preliminary results now, so I won't be showing them, but an expert survey found almost identical results in terms of levels of support. So in closing, conifers have persisted, of course, for hundreds of millions of years, and they have locally adapted since the last glacial maximum over a wide range of conditions. And this is despite their relatively slow molecular evolutionary rates and their slow rates of migration and capacity for migration. Now, standing variation for climate-associated traits appears to be quite high in the species we've studied, and that is certainly good news for long-term climate adaptation. It doesn't mean that populations are going to be able to adapt quickly, but it probably means that in every population there are at least some individuals that can handle projected future conditions, as long as the population remains within the climatic niche of the species. Another bit of good news is that phenotypic and genomic approaches identified similar patterns of local adaptation and similar climatic drivers. And I think we really are going to need to use those climatic drivers to design reforestation strategies for new climates. So identifying what climate variables the populations are most closely adapted to is facilitated either with phenotypic or with genomic data. I think there's a big need to branch out from just studying the hosts, that is the trees, and move into more genetic and genomic studies of the pathogens and also insects. And I think the work that Richard Hamlin's group is doing on Dothostroma needle blight, and they're also working on the pathogen that causes Swiss needlecast and Douglas fir. I think that kind of work is really important. We're integrating that work with the work on trees by building risk models of climate change, that is the risk of the abiotic factors to trees, but also the risks of these biotic factors in terms of how they respond to climate change. And I'm quite excited about bringing all those pieces together at the end of CoAdaptTree. And finally, in Canada, I don't know about in Europe, but in Canada, social acceptance of assisted gene flow as a reforestation strategy is high. We have moved to assisted gene flow, or what is called climate-based seed transfer in British Columbia. That is now in forest policy, and I think we will see similar changes. However, I do want to emphasize that we need to make small moves over time, because these trees are most closely adapted to low temperatures, and we see population variation that is quite strong in cold injury. We do need to be careful not to move things too far, too fast. So I think we still need to do some very good thinking about how to balance the risks of moving against the risks of not moving. And with that, I would like to thank the many, many people who have been involved in this work. This photo includes people in my immediate lab group, but there are so many people that have been involved with AdaptTree and CoAdaptTree, and I just really appreciate all of their work. We have also had funding from Genome Canada, Genome BC, the British Columbia government, other genome centers across Canada, as well as a variety of academic institutions and forest companies for this work. And now, hopefully the video conferencing will work and I can answer your questions. Thank you.
Yes, I can hear you loud and clear. How do can you hear me? Yeah. Good.
Very good to see that this, yeah. Very good to see that we can.
Hello, everyone.
Have you.
I wish I was there.
But it's great that we have a chance to have your talk here, and then that you don't have to travel all the way, but we can still take questions. So please, other questions to Sally? I can start with the first one. I was actually curious about the, when you were explaining in the beginning, why actually is the adaptation to cold hardiness so much more pronounced than the adaptation to drought tolerance? You had the first...
I can't quite hear the question. Is there maybe a microphone or?
The question was, why is there so much adaptation to cold tolerance and not to drought?
Yes, very good question. And who asked the question?
Marcus Lindler.
It was me, Marcus.
Okay,
Hi.
thank you. Thank you. Very good question. It's something that I'm very curious about. I think there's a few possible explanations. First of all, we're talking about a species that exists over areas with relatively short growing seasons. And it could be that, and we saw this in lodgepole pine, we saw this in interior spruce, but we also see somewhat similar pattern in Douglas fir, which comes into quite a wider range of growing season length and drier conditions. So possible explanations. One is that these conifers are globally pretty adapted to drought, but they're not locally adapted to drought, and that one level of drought hardiness really deals with the range of conditions they find within their ranges. Another is that local adaptation to drought is on such a fine spatial scale that, given the scale of gene flow, you don't get local adaptation occurring because the gene flow swamps that highly variable local availability of water. A third is that our sampling strategy is one that picks up big patterns, but it doesn't pick up those fine scale patterns. So I think those are all possible explanations. Even though we've seen now this result in I will say 2.5 species, because the Douglas fir, the two varieties differ quite a bit. There is some variation among some populations in the Douglas fir. I think it's something that we can't ignore drought, but there's no question that the overwhelming signature that we see in all of these species is that adaptation to low temperatures. And we're still pursuing drought. We've got studies underway in another species this year, in Larix occidentalis, and hoping to also work on Thuya plicata.
We have another question that you will come into the computer.
Okay, great.
Good morning, I hope it's coming at this point. I have a question related to how much do you believe in breeding to improve resistance to biotic agents? Because you are talking about Dotristoma, but we have seen, well in Canada that it can be a bark beetle, the problem. It can be very diverse biotic agents. So how much breeding can help to solve that?
Yeah. So I think that breeding for resistance is only going to become more important in breeding programs. The programs in British Columbia have put an increasing emphasis on health and health-related traits. Some traits are difficult to breed for, like mountain pine beetle resistance, just because it attacks trees that are older, and the amount of time that it takes to run through breeding programs is pretty challenging. But certainly Dothostroma is starting to be under selection in some breeding populations for lodgepole pine. And I think that with climate change, there's two components here, because of course we're seeing expanded ranges and expanded impacts of a lot of insects and diseases. And the second piece is a stressed tree is of course more vulnerable than a tree that's not stressed. So much of our work relates to that abiotic stress factor. But I think that selection within populations for higher resistance is certainly something that's very valuable.
Yes, one more question.
Maintaining diversity similarly is very important.
Please come to the front and ask your question here.
Oh, hello, Carola Mosnofidu from FOR Freiburg and BOKU Vienna. And I would like to ask you whether this cold hardiness adaptation rather reflects altitudinal or latitudinal patterns, or whether we have similar patterns along altitudinal and large scale scales. Thank you.
Yeah, so that's a very good question. And it's quite interesting. So our topography is very complex. We have our coastline. So we have patterns that are somewhat longitudinal from the Pacific Ocean inland, but the coast runs at an angle. It doesn't run north-south. And then we have a lot of mountains. We have many mountain ranges that create different patterns as you move in from the Pacific, from the west to the east. And so the patterns are pretty complicated. However, it's interesting that at one point there, I went through fairly quickly maps of those two clusters, so cluster 4 and cluster 6, that were providing, one was providing variation at the very cold end of the range across the Rocky Mountains, the East Mountains, and the other was providing more variation among populations west of the Rocky Mountains. It's interesting that one of those clusters, I think it was cluster 4, shows a similar pattern of change in average allele frequencies within that SNP cluster with latitude and with elevation. So you're seeing the same SNPs increase in frequency as you go north or as you go up in elevation, and that's something we might predict. But that other cluster doesn't show that pattern. You don't see the same pattern with latitude and with elevation. So I think that I think that what we're finding is these patterns are very complex, and that it's difficult to make those sort of broad generalities about the patterns you'll see. I think that's different in some geographic places, probably in Finascandia, where you have climes that are fairly one-dimensional. You're going to find more consistency.
Thank you, Sally. As you were getting up at 5:30, I will allow one more short question, and please a short answer as well.
Thank you. It's Tor Myking. And I was wondering, for which kind of cold do you have these associations? I mean, is it absolute cold? Is it even summer cold or winter, or what kind of?
Yeah.
Cold? Thank you.
So it's, on one hand, it's difficult to sort. So we phenotype cold hardiness in the fall. We focus on fall cold hardiness there, but we also record bud phenology. And really, the timing of bud break is highly correlated then with the loss of cold hardiness in the spring. So in the fall, we see these fairly strong patterns among populations, and stronger than the variation we see in the spring. And I think Ian McLaughlin showed you some of his data on that this morning for you. And so when we look then at the climate variables, we see a whole bunch of cold related variables, but those are associated with cold, but they're also correlated with each other. So it's hard to separate. Mean coldest month temperature in winter always comes up. The length of the growing season comes up. Frost-free period. Mean annual temperature often comes up. Extreme minimum temperature. So we have a modeled climate variable, which is 30-year extreme minimum temperature, and it's highly correlated. So I think that the colder the environment, the earlier the trees set bud, the earlier the fall frosts come, the shorter the growing season, and we can't sort those things out. But the important thing there that we need to keep in mind is that if we're using assisted gene flow for reforestation, which we are here, we have to be careful not to target too far in the future, because we're going to run into cold hardiness problems.
So thank you very much, Sally, for being available, for answering our questions. This was really excellent. It worked very, very well here. So thank you very much. And a warm applause to Sally one more time.
Thank you very much.
We have recorded this question answer session, so you might hear yourself again on the video we're going to produce in about a month.
Okay, great. Thanks. And I wish I was there. Thanks for letting me do this by video.
And thanks for being awake at 5 o'clock in the morning.
No worries. Bye-bye. Enjoy the meeting.